Last morning with me? Wow, that's a lot. Ok, thank you. Penso che sia di più per quelli in cui siamo in streaming che per quelli in aula, che tanto siamo... Mi sentono bene. So today we'll discuss a little bit more subsurface. We'll go into details of hydrogen metabolism by microbes. I'll show you a few approach, and at the end of it, I'll show you a decision diagrams, which I think I developed some time ago when I was presenting at the Italian Geochemical Society, which I think could be interesting for you to see, because that's how I think we should approach the interaction between other disciplines involving underground hydrogen storage and microbiology. First of all, subsurface environment, okay? Because all our storage right now is in the subsurface. So it's only fair if we discuss a little bit more what microbiologists see when we see subsurface ecosystem. So at the moment, subsurface biosphere, so life in the subsurface, is loosely defined as the region beneath soil and sediments where the limits of habitability are defined by physical processes. Most typically, this is temperature. So we define the habitable zone in the subsurface from the surface or near surface sediment and soil, all the way down to temperatures of 130, 135, depending where you want to put your RT limit. We know we can grow things up to 122 in the lab. We know the environment might be a little more. But you could say we go from the surface of the sediments all the way down until we have a certain porosity or connectivity of pore or water activity. So you can define it in different ways. Right now, most people will say you are in the subsurface when you pass about a meter depth in the soil or sediments. So already at 2 meters depth in sediments, you are considered in the subsurface. This is because the exchange with the surface in marine sediment, for example, is limited by diffusion at the top portion of the sediment and by burrowing animals. So things like worms and clams that make a hole in the sediment, they will allow oxygen to penetrate. Typically, these animals, they don't go deeper than 30 or 40 centimeters. So when you are beyond that, there's little communication between what is in the sediment and what's in the water column at the top. Similarly with soil, it's difficult to get influence from the atmosphere and winds and things like that beyond 1 meter depth in the soil. It's a slower process, the interaction with the surface at that depth. So most people will say from 1 meter depth into soil or sediments down that subsurface environment. Now, for the purpose of hydrogen storage, we're most likely talking about hundreds of meters to a few kilometers. So we are well within this definition of subsurface ecosystem. We know now that the biosphere is very large in the subsurface. It contains about 70% of all bacteria, archaeals there, and possibly over 80% of all the known species are down there. So microbes, prokaryotes, are by far the most abundant organism in the subsurface, and probably the subsurface contains the highest diversity. Now, you might have heard people talking about the Amazon rainforest as the place where you have the highest diversity of insects. So if you want to find new insect, you go to the Amazon, you're very likely going to find one. Where for microbiologists right now, if you want to find new microbes, you just dig, and you're going to most probably find new things nobody has ever seen before. We have average estimate that suggests that we have 10 to the 29, 3 by 10 to the 29 cell only in the subsurface. So surface environment add to it in cell number, but not a lot. The majority is actually in the subsurface. This volume of the subsurface ecosystem is huge compared to the soil, to land, to even the ocean. So even if cell number are lower in the subsurface compared to seawater or soil, the volume is so big that things become huge very quickly. Early investigation of subsurface biosphere started really around coal beds and oil fields in the 1920s, 1930s. But we did not start really to speak about subsurface biosphere until this paper here, 1992. Thomas Gold, which was a geochemist, a geologist, he had controversial theories about the abiogenesis of petroleum, for example. He also had a few brilliant ideas during his life, and this is one of these. So this was published in the Proceeding of National Academy of Science, PNAS, and it titled this paper "The Deep Hot Biosphere." In his word, since we have energy supply that comes from chemical disequilibrium within the subsurface due to rock types, due to hydrothermal system, due to imbalance and the activity of the planet, there might be a subsurface biosphere. At the time, he spoke about a hot subsurface biosphere because he was thinking that where you have thermal gradient, you have a higher amount of disequilibrium. But right now, we know that it's not present just in hot places, but it's present in many other places. And why we know it? Well, we've been drilling all over the place with deep sea drilling program, and then the ocean drilling program, and then the International Ocean Discovery Program, IODP. You may be familiar with it. We've been drilling all over. We've been doing it on land also. This is the example of a continental drilling operation in Oman drill site. These are ophiolites that were drilled into. But we've been drilling through every major rock type and lithology around the planet. And every time we do that, and we involved microbiologists, not just geologists in the drill site. Oh, the other things we do is getting access to deep mines, right? So these one are Canadian deep mines, but people were working also in diamond and gold deep mine in South Africa, for example, and in other locations globally. Yes. I don't know which mine is this one. This is one of the pictures of where they found isolated brines that were 1.8 to 2 billion years old in Canada. I'm not sure what the name of the mine, but it's probably that one. This is work by Barbara Sherwood-Lollard and her team on finding these isolated brine pockets, sampling them and demonstrating the microbiology. It's been isolated but very active there for the past 1.5 to 2 billion years. Yes.
So in all the different environments, hot, cold, salty, and everything, is there any affinities of microbes that you can?
What do you mean for affinities?
No, I mean like the amount of microbes you can find, or the variation of microbes. Like, will you get more variation in thermal areas?
Well, we see that at high depth, for example, microbes are more abundant and more active when we find organic rich layer. So if you approach a coal bed layer or a sapropel within sediments, then you see a spike in microbial abundance and activity. Makes sense. There's more organic carbon. That's food for heterotrophs. The microbes, they use organic carbon as carbon source, so you find more of them. We also find more microbes and more activity at the contact between different lithologies. Why? Well, you have different chemicals between two different types of rock, and at the contact point, there are more chances of getting good redox chemistry going, right? So if you think about what we discussed yesterday, every life on the planet needs an electron donor, an electron acceptor, and some carbon. You can answer yourself every time you think there are good imbalance in redox chemistry, every time you think there are good carbon sources, inorganic or organic, like enriched CO2 circulation in aquifers within carbonate system, for example, then yeah, you find plenty of microbes. But there is a lot of variation. In spite of the variation, there are some parameters we can use to estimate cells. So we do drilling, we do deep mine access, and this is what we figured out. This is number of cells, prokaryotic cell, bacteria and archaea, by different environment, underground, soil, oceans, animal guts. We hear a lot about human microbiome and other animal microbiome in the gut, like being a huge ecosystem, is a tiny dot as total number of cells globally by the total number of animals compared to the subsurface. And if you put together a lot of the drill sites, you see those in the Pacific on the right, and they're colored by where we are in the Pacific. You see this is number of cells on the x-axis by depth on the y-axis. And you can see that things get less abundant as we go down in depth into sediments. And yes, the slope of this decrease in cell number is different in different locations globally because it changes based on the amount of organic carbon that is present. For example, the yellow dots there are from the South Pacific gyre. oceanographers know very well that the South Pacific Gyre is an area where there's very little surface primary productivity. Waters are super clear because they're limited by nutrient and iron, so there's very little biomass produced at the surface, which means there's very little carbon that sinks down to the bottom of the sediments, which means the sedimentation is very low, and burial of organic carbon in sediments is very low, which means there's not a lot of food. So cells are by far the least in number compared to the dark places, the black dots, which are coastal environment or high productivity environment, in which we see order of magnitude more cell, 3 or 4 order of magnitude more cell at the same depth into sediments. But we've been doing this job on land and at sea. And right now we have cell counts that are up to 2.8, I think, to 3 kilometers deep in continental system and up to 4 kilometers depth, 4.5 in marine system. So we've been able to count things pretty deep. We see there are cells. We see they're active because we do experiment and we see they respond to input of organic carbon and other things. We don't know where they stop. For sure, when your temperature gets too high, but there are locations that have been modeled, like the Marianas Forarc. I don't know if you're all familiar with the term forarc. It's basically where two plates collide, one goes under, and then the portion you have immediately at the contact with the two plates before you get volcanism and other things is called forarc. So it's the bending of the one plate on top of the other. In this portion here, where the plate is going down, the plate is essentially a big block of basalts and sediments that is relatively cold, depending how old is it. In the Marianas Forarc, that plate is very, very old. It's one of the oldest on the planet before sinking. So even as it is sinking down, the majority of the kilometers thick basalts, gabbros, and sediments stays at a cool temperature for a long time while it's going into the mantle. And people modeled the 122 line temperature limit for life to go as deep as 20 kilometers in that area. Now, because, and we go back to this, if you're limited by pore space and subsurface, what matters is your rock type, right? You are in highly crystalline rocks. If you're limited by temperature, which is the other parameter that has a hard stop somewhere between 122 and 135, 140, then you are limited by heat gradient. How hot is the area where you're in, right? If you are in a very cold area where the heat flow is very low, you can go a lot very deep. Cantiflegrei out here, which is a place where you have a very high heat flow, where in the center, where we will go today, few centimeters below the soil, you get to boiling temperature, essentially. You know your life doesn't go anywhere. It's actually just on the surface, right? Because you are boiling immediately under. So heat flow is very important. We'll see about that. Yes?
Just a side question. Why did you say that the maximum temperature is 135, 140? You don't believe that there's life above that?
Well, we have no evidence, okay? Our laboratory evidence says 122. Now, we know because we are not very good at growing things, that if we can grow somebody at 122, there might be something higher. We don't know how high because we never seen it outside. It's also very true that right now it's difficult to prove the absence of life out there. Like if you go in a place, you run out of detection limit for whatever technique you use long before you can demonstrate that there is no life there, no cells. But there are theoretical constraints that people set forward right now, like the stability of phosphate to phosphate bond, which are fundamental bond in every life on this planet, because we store energy in something called ATP, which contains 3 phosphate bonds, and that's where energy is. Now, these bonds are stable above 135. So people say, well, that might be the limit. We've been wrong before in the past, because in the past we said, well, the limit is DNA decoupling above 75 degrees. And now we are at 122, because life found ways to deal with that. So we don't know where the hard limit is. Right now in the lab is 122. Many people use that as a hard limit in the environment, but I will keep it a bit more loose and a bit higher because that's a laboratory limit. So most of my colleagues will agree that we might be anywhere between 122 and 135 or 140, somewhere in that range. And if you want to be sure, move it to 150, above 150. So you are definitely sure for now that you are far away from whatever evidence we have. We might still be wrong, but at least you are far away from whatever evidence we have today, right? We are based on data. So our data is here, we put it just a bit up to have some level of confidence.
So, no life at a no cell at all, you know?
Yeah, no cells. You don't see cell, you do not measure cell, you do not see activity linked to cell, like using stable isotopes or radioactive isotopes or things like that. But that also might also be limited to the techniques you're using. That's why people that try to see if there is life use like 10 techniques, all different. And every technique turns out negative, they say, well, probably there is no life here. But still, we know every one of these techniques has a detection limit, right? It's like in geochemistry and chemistry. You go in the environment, you measure something, you can never say there is zero. You can only say there is below detection limit. Then you can assume that, you impose that to zero, but for all purpose intended. But in reality, you don't know, because your detection limit is not zero. It's something higher than zero, right?
Is it the same with virus?
Same with viruses. We don't know when they stop, and viruses are even more difficult because they're smaller, more difficult to count, and you never know if they're active unless you see them reproducing inside the cell. So to see virus, you need to see a cell to test if they're active. Now you can find the DNA, but DNA is stable also a higher temperature if it's bound to organic matter. So it might be just dead things you are seeing with DNA. You need to demonstrate that they're alive. It's like saying, oh, humans are living like now in glaciers. Why? Because I found a dead body frozen. Okay, the dead body got frozen there, but nobody's living there. You need to demonstrate that there is activity, ongoing life, to say there is habitable, right?
So how do you do that? How do you demonstrate?
For example, incubation with different substrate. You get a sample, you throw inside some food, electron acceptor, electron donor, sugars, and you see a bloom of activity. Say, yes, there are cells, they are responding to my stimulus. That's one way to do it, for example. There are other ways. There are ways you can color cell to check the integrity of the membrane, for example. Because a live cell can maintain a gradient between outside and inside. It can avoid things to come in. If you are dead, things just penetrate. So there are ways of coloring cell, assuming you can see cells, that you can color to discriminate intact cell membrane from broken one. And there are a series of tests like that. We have maybe 15 or 20 ways to check that. And then it's up to you how many you want to test. So this is a paper that was published some time ago in 2012, where people got every cell count that was out there. Now we're zooming just in a few of them, but what they did, they related the cell count to the lithology of marine sediments and to the amount of chlorophyll that we can measure from satellite, primary productivity in the ocean, and they calculated where cells are on this planet. And then this work has been redone in 2018, including marine and terrestrial ecosystem, and that's how we get to the estimate. And people in terrestrial ecosystem use the rock type, available pore space, heat flow gradients, amount of organic carbon in the different rock type to calculate how many cells we have there. So we have constraining parameters that help us to estimate how many microbes there are in the subsurface in different locations of the planet and at different depths. And we know exactly how fast cells are decreasing normally. There are surprises though in this. A big one comes from marine ecosystem. It's called the sulfate methane transition zones. So this is a zone defined by geochemistry in marine sediments. Now, if you go out here, or you go out in the middle of the Tyrrhenian Sea or the Atlantic, and you start to drill down, and you start to measure the concentration of sulfate that comes from seawater, sulfates will start to go down in the sediments. This is because microbes are using it as an electron acceptor. They're doing what is known as sulfate reduction, right? They keep doing it until sulfate disappears, and around the area where sulfate is going to zero, methane starts to increase. Because the community has depleted electron acceptors, CO2 is the last one, hydrogen is produced by fermenters. You remember, fermenters are the microbe that can break down organic matter without using any electron acceptor. They dump electron on an intermediate. So hydrogen is a byproduct, and microbes are doing H2 plus CO2, and they make methane, methanogenesis. Now, that area there is an area where there's a very high microbial abundance and activity in marine sediments. This is because you get an electron donor from the bottom, methane, that is coming up because it's a gas, right? So it's going toward the surface, it's diffusing toward the surface. At the same time, you get sulfate coming down with the seawater that has been trapped in the pore space. And microbes have in that area called SMTZ. They have a good electron donor and a good electron acceptor. And there are microbial consortia that work together where one guy is doing metal reduction, the electron is transferred to the next guy in front of him, and that guy is using the electron to do sulfate. Sorry, metal oxidation, transfer the electron to a guy that is doing sulfate reduction. So they work in couple, symbiosis. They don't do the entire things themselves, they actually pass electron between cells. Yes?
Where does this hydrogen come from then? For the hydrogen
Fermentation mainly. So degradation of organic carbon by microbes that can break sugar into pieces. And then one part of the sugar is consumed completely, the other half, which is called pyruvate, to half, you dump electron on it and you use it as an electron acceptor. Fermentation is the process that allows you to use sugar to produce ethanol, alcohol, in your beer. Fermentation is the one that produces lactic acid from lactose in yogurt, kombucha, tea, fermented teas, chocolate, coffee, bread. They're all type of fermentation. Wine. Those are microbes that can use the sugar, but instead to burn it to CO2 and only, like we do, because we use oxygen as electron acceptor, so we're able to break down sugar completely and dump electron on oxygen, they stop midway and they release a product outside, which is usually lactate, acetate, propionate, ethanol, a number of intermediate, partially oxidized, partially reduced carbon compounds. And one of the side products is hydrogen, because when you don't know where to dump an electron, but you have the right machinery, you have protons around, you can just dump it on protons and make hydrogen.
How does this SMTZ zone correlate with the carbon compensation happen?
Oh, we are beyond that. We are in sediments, it's everywhere. It doesn't matter where you are depth-wise, once you are in sediments, you have an SMTZ. The beauty of this SMTZ is that the depth in sediments, we are depth in marine sediments, changes from hundreds of meters deep inside sediments to centimeters deep. And depends on two things. Porosity of the sediments, because you want to limit the penetration of oxygen and seawater, and so you get anoxic very, very quickly. You start to consume sulfate and make methane. An amount of organic carbon. If you are very organic rich, things get used, consumed very fast. Fermentation kicks in very fast. Hydrogen is produced very shallow. So you can find the sulfate metal transition zones when you go a few tenths of centimeter to a meter in coastal, very fine sealed type of sediments, or hundreds of meters deep when you are in open ocean, no productivity, very coarse type of sediments. This is just to tell you that there are these horizons in marine sediments where just microbial activity is creating these horizons, right? This is not an horizon that is made by lithology or changes in rock type that is providing electron acceptor and donor. This is microbes consume something, suddenly somebody else can produce something, and the interface between the two, you have both electron donor and acceptor. Now, tomorrow we do a practical together, building what is known as a Wiener-Grasky column, which is a microbial ecosystem. You bring this with you, they last tens of years, they keep growing and changing. In those systems, you start out with a completely homogeneous inoculant, and then by the few months, you get completely different color layer that are suggesting that things are being exchanged up and down exactly in this way. So this also goes back to the question of David yesterday. Can we assume an homogeneous reservoir and get? It's never homogeneous. Even just gravity is going to create gradients, right? Of gases going one way and heavier stuff going the other way if you are in liquid medium. And that creates gradients. And if microbes exploit the gradients in a different way, then you get suddenly you get transition zones where things are possible. So in totally homogeneous marine sediments, just by compaction due to gravity and depth, you suddenly find a layer where you have higher microbial diversity and activity compared to above and below, okay? So the point here is not so much that you need to know sulfate metal transition zone, but it's what this can teach you about how microbial ecosystem work and interact. Any other question?
Yes, Renato. So essentially, you have to take this kind of diagram with the geothermal temperature to get the distribution of the gas hydrate.
Yes. Yes.
You know what are gas hydrates? The portion in the marine where you have the solid gas rate or solid gas methane.
Metal is trapped in methanized, basically.
Does it exist a map published in the values of CM of the sulfate methane?
I don't know if there's a global map, I need to check, but in IODP cruise report, we always have the position of the sulfate metal transition zone. So it could be drawn on the map if it doesn't exist. Take notes, that's a good one to make.
Because I got the map of, for example, in Tanzania, we wrote a small paper where we show the position of gas gray using the siphons. Because we got this weird reflector which is simulating the sea bottom. But we don't know if there are data of those types in there.
If you have any drill site, we can figure that out. Yes.
Since this is geochemically defined zone, can we do the same with other pairs of oxidized?
Absolutely, yes. You can define as many transition zones as you can measure. You can define the transition between H2S and iron 3, where H2S is coming up, there is a good electron donor that is generated by sulfate reduction, and then iron 3 is going reduced to iron 2, because they're meeting and microbes are they're doing H2S oxidation to sulfate or elemental sulfur or thiosulfate, depends, coupled to iron 3 reduction to iron 2. So you can define as many ingredients as you want. This is a very important one. Oh, well, this is a very well studied one. That's why we know it. We know it very well. But any transition zone is a good. Microbes like geothermal, geochemical, redox disequilibrium. So every time you have a change between these two things, you are in a zone where there's plenty of food to make complex things. Any other question? One big misunderstanding about microbes is the scale at which microbes work. Okay, so we said yesterday, be mindful of the scale at which microbes experience their environment, right? We said life and low Reynolds number. Things are like a gel, diffusivity changes in the way it behaves when you are in very low Reynolds number, interaction with things, swimming is not swimming, is more like crawling through a gel and things like that. At the same time, when we start to think like that, we think the microbe interact with things just at the microscale. Because of their sheer number, effect of microbes are huge on very large scale. This is an exercise we did some time ago, thinking about processes that are present in subduction zones. So again, one plate going under the other. We were thinking about a specific place. And we have spatial scales from micrometers to hundreds of kilometers, and then temporal scale from seconds to millions of years. And now we mapped biological and geological processes. And you can see that the blue and the yellow, the orange diagram, they overlap in a lot of places. Because if you take, for example, microbial effect or mineral precipitation and dissolution, you see their effect in the environment on scales that go from centimeters to kilometers. And that overlaps in spatial scale with things like phreatic eruption, hydrothermal ceilings in volcano, landslides, and things like that. At the same time, you can go on the microbial scale, on the temporal scale. I mean, the oxygen we are breathing right now, 20%, 21% oxygen, is in large part due by the activity of microbes in the ocean. The oxygen you're breathing right now is not being produced by the plant out here. It's most probably being produced 10,000 years ago by microbes somewhere in the Atlantic Ocean or nearby. So microbes are small, but they interact with geology at very large spatial and temporal scale. And because of that, you have big interaction. One example, when we started digging around, we started measuring cell abundance in sediments, right? sediment depth, centimeter below seafloor. And then you have, this is sulfate reduction measured for each cell, femtomole of production. So basically, they've been measuring the activity by sulfate reducer, which are an important group. And then you see calculated biomass turnover time. How long it takes for microbes to double in size, essentially, to change all the cell, right? Population measurements. And you can see that it goes from a few months at the top to 10 years at the bottom. At 50 centimeters, cell double in number, they turn over in 10 to 15 years. But it's even better. If you look at the sulfate meta transition zone in some area, for example, here, we are at 2.9 meters depth. Or you go and you convert this one into sediment age. And you look at the number of cells to assume steady state, to assume the cells are always the same number, they're not just declining over time. You get generation time, that's a log scale of 10, 100,000 years. And in some place, people calculate that if microbes are doubling, we know they're there, we know they're active. If they're doubling every 1,000 to 10,000 years. Now, which means that's one generation, 10,000 years. To get multiple generations going, you are in geological timescales. You're thinking about hundreds of thousands to millions of years. And there's an interesting paper, which I like you to read, which is called Time as a Microbial Resources. Just 2 pages by a colleague from the university. She was in Tennessee, now she's in Southern California, in LA. So she discusses time as something we usually do not consider. Now, she uses an analogy for this. I mentioned it briefly yesterday. We tend to think about time in human scale, right? And for us, things that happen on very long time scale are almost non-existing. It's like an ant born in the summer and an ant born in the winter. When they come out and they see a tree, that tree is stuck in time. It's either with leaves or dead. Because they last a few weeks, they don't know that a year later that tree is going to lose the leaves or get the leaves. Because their time scale of measurements is completely different. If there are microbes in sediments that are hundreds of thousands to million years old, and these microbes, we can stimulate them and see that they're still alive and probably active. Those microbes probably have been interacting with that plate for a long time. One of my colleagues is very provocative. Some people say that a microbe, think about the ocean, okay? You got the mid-ocean ridges that are pushing the plates apart, right? They're growing. And then plates after 100 million years or so, they fall down and they go back into the mantle. That's the cycle, the Wilson cycle going, right? A microbe deep, 2, 3, 5 kilometers deep into the sediments needs to grow. So if the plate is going that way, one way to hell, into the mantle, needs to grow on the other side, just at the speed that the plate is moving. So the plate is moving 1 millimeter a year, microbes need to duplicate just 1 millimeter a year in the other direction to stay stationary. So he says that microbes are still, a plate moves through them.
Hotspot, like the hotspot.
Like a hotspot, microbial spot, the same way hotspot work. Plate move through microbes. Microbes just need to beat how fast the plates are going. And we know there's a lot of colonization out there in plates. Yes.
Right. So you say a single cell can live 10,000 million years.
Well, that's what we estimated based on activity, integrity, and a lot of other things. Some people call them Methuselah. They've been alive for a very long time, this cell. Some people think these cells are just resting in a dormant state. They're hibernated. So it's not like they live metabolizing all the time, but they live suspended in time until they get a pulse of something. Then suddenly they start to life again, and then they fall asleep for a very long time. Like tardigrades on masks in museums, that they stay dormant for 200-300 years, and then you can revive them.
Then how do you test for that? How do you say that?
Well, you see that they have membrane integrity, you see that they have energetic
Not
requirement.
the age.
No, you don't test the age, you test how much energy they're consuming. And you know how big is a cell, and you know how much it makes to make another cell. Then you say, okay, if they're getting femtojoule of energy every day, to make a cell, you need millijoule. How long does it take to accumulate that energy down here? Well, so it'll be at least 1,000 years, right? So maybe it's not like they're getting that energy every day and then they build the cell every 1,000 years. But on average, to be steady state, every 1,000 years, you need a spark of something to make them spore. But they're for sure sitting there for 100,000 years because there's not much connectivity. You are buried 4 kilometers deep somewhere.
That means you're investigating its metabolism.
Yes, nanocalorimetry is one popular technique to measure the activity of very low activity of dormant or semi-dormant cells. So now let's move. Now that you know that there are a lot of cells, they roll, they interact with geology, they create their own geochemical gradient, they react to geological gradients. Let's move a little bit back to hydrogen, okay? Yesterday we spoke about carbon, nitrogen, sulfur, iron. We looked a little bit at the metabolism. You're going to be able to read more once we give you the slides about the different metabolisms. Hydrogen is an interesting one because hydrogen is considered the universal electron donor. Why? Very reduced. The only thing he can do is give electron, and he's very happy to give electron. Why? Because the nuclei is very weak, and so electrons jump out very, very fast. And molecular hydrogen, which has 2 protons with 2 electrons floating around, anybody that comes around that has a little bit of electronegativity is going to pull that electron away. So hydrogen, when it's present, is a fantastic electron donor and can give electron to every possible electron acceptor that biology can use. So to oxygen, to nitrate, to iron, to CO2, to all of them. You name them, okay? The enzymes that interact with hydrogen are super interesting. You remember, enzymes are proteins that catalyze a reaction, right? And a special class of those catalyze redox reaction. So stripping electron from hydrogen moves the oxidation states from 0, H2 to H plus 1. That's a redox reaction. Requires enzymes that are able to get the electron from a very good donor and then pass it gently somewhere else. Okay? And you want them to be as good as taking the electron first before hydrogen bounces off, giving electron to random things that are sitting around. So they need to be powerful and yet capable of moving them around. Microbiological analysis told us that we have a very large diversity of what we call hydrogenases, which are the enzyme that uses hydrogen. And they're very different. They can use H2 and make H+. They can use H+ and make H2, so they produce hydrogen instead of consume it. They can sense it in the environment. They can bind it. They can control it. At the heart of it, we think that hydrogenases evolved many, many times. And they're distributed almost everywhere in the tree of life. So if you remember the big diversity of bacteria and archaea that I showed you, a lot of those have hydrogenases around. And microbiologists speak of hydrogen as the microbial energetic currency. Everybody can use it. Many can produce it. So it's just exchanged constantly between cells.
As you were saying about evolving, I had a question like that relates from the video that you showed yesterday, how the different microbes related to different cells. Can we, like, if we extract any of the bacteria at any point of the gel, can we trace back their evolution?
Oh, in that case, that's what they did on the paper.
Okay.
They know exactly what mutant comes out of from who and what's their lineage. And then they sequence the genome and compare.
Yes,
That's
so they don't lose out on the previous DNA.
No, you come out with a copy of your previous person with few modification. Copy with modification, yes.
But you just consider energy exchange currency and you said they consume it as where should be then?
No, they consume it. They consume it, they produce it, they sense it. Hydrogenases are used to do H2 to H+, H+ to H2. There are hydrogenases that can do everything with hydrogen. It's consumed by a lot of cells, it's produced by many other cells. And even cells that we would not think they're exposed to hydrogen, they actually use hydrogen a lot. One place where hydrogen is fundamental, for example, as a microbial currency, is microbes in your gut. You produce a lot of hydrogen every day. And a lot of that hydrogen is converted to methane or used in other things. So you have microbes in your gut that produce hydrogen from your food and then exchange hydrogen with everybody else. And hydrogen is also a powerful signaling molecule for interaction between microbes and eukaryotes. It's so important that the enzyme that can use hydrogen evolved many times. If you think about photosynthesis, right now our planet functions because we have oxygenic photosynthesis out there. Plants and algae and small bacteria, cyanobacteria that make oxygen and produce biomass. That's the base of the food chain on the surface of the planet everywhere. And yet oxygenic photosynthesis evolved only once in the history of this planet. And it's the most important surface metabolism. Hydrogen evolved maybe 15 to 20 times because we have completely different lineages of hydrogenases that evolved over time and emerged over time. Now, there are, we call them hydrogenotrophs. Trophs from eating hydrogen, no? Microbes that eat hydrogen, or hydrogen oxidizers, because they oxidize hydrogen. Now, as always, in this case, you can have an aerobic one, meaning that they oxidize hydrogen at the expense of oxygen, or anaerobic one. They oxidize hydrogen at the expense of everything else besides oxygen, right? Very anthropocentric division of the two. All the hydrogenases we know out there, and this is a figure made by Guillermo for Martina's paper, all hydrogenases we have out there, they fall into three only categories, depending on the metal center they use to strip electron from hydrogen. They're either nickel iron, so they use that. So these enzymes, these signs around here, are sulfur from an amino acid, cysteine. There is binding to this nickel and iron cofactor, which is the catalyst that takes the electron from the hydrogen, or their double iron center, iron, or single iron. Now it's interesting because biology evolved these three metal centers to be able to strip electron from H2. And if you read papers in engineering, where people are trying to get better catalysts to make hydrogen or interact with hydrogen, one of the best catalysts known to interact with hydrogen is nickel, in nickel iron alloys. So we are rediscovering that to interact with hydrogen, we need exactly the same metal that biology has evolved 3.8 billion years ago or so. Because we think hydrogenases were among the earliest enzymes that evolved on this planet. Now keep in mind that diversity is huge, but usually you either a nickel iron, iron, or iron only enzyme. That's the metal center that interacts with the hydrogen. And then they become very diverse. It's another figure from the same paper. Like you see the nickel iron hydrogenase, they're very different in shape and size. They're different in how complex they are. They're different in the way they interact. But at the end, you see, this is a nickel iron center. And then there are other metal, iron sulfur in this case, that is lined up for electron to jump around and then be passed away, like we were saying yesterday, right? Proteins are complex 3D shapes that keep things in the right place. Redox protein are complex 3D shapes that keep metal center in the right position for electron to jump around. It's like, shapes and function are related as they often are in engineering and in design. It's the same in biology. Shape and function are very related. So, yes. Okay, in this case, within the same hydrogenases, the different color code for different proteins that are attached together, because these are multiglobular proteins. So they have different parts that are assembled together, and colors are assembled by part. Some of them are two identical parts, like this one. This portion here is identical to this one, and then this one is identical to this one. And it takes 4 of these globules matched together to make the entire machinery. It's like, when you're building a robot and you give him 2 arms on one side or the other, symmetrical, specular, that's what happens very often in proteins. We call this quaternary structure. From the simple list of amino acids folding, refolding, and then assembling multiple pieces. So simple list of amino acid, folding, refolding in complex globule, attaching multiple units together, quaternary. It's complex final structure. So color is just telling you what things are preserved. For example, this one, two identical specular pieces together, same color, means that you have two pieces of the same attached face to face together. Now, for you, it's important to remember that hydrogen can be used and produced by biology, used and produced by biology. The enzymes that can do it are very, very diverse and emerged multiple times, and they're present almost everywhere in the tree of life, and they evolved very, very early in the evolution of this planet. So it's a very important electron donor, and it's considered metabolic currency. What do I mean for metabolic currency? Let's go back to our diagram of the subsurface, where we were going deep into sediments, right? And we said that at the bottom, where methane is produced often with the use of hydrogen, right? Now, if you go down there and you measure free hydrogen, okay, you take your sediment, you squeeze it to squeeze out the pore water, then you take the pore water, you extract with headspace extraction, inject into the GC, and you go measuring the hydrogen. Hydrogen levels there are nanomolar to picomolar. It's very, very low. You see almost no hydrogen in the waters. But if you look at cells and what machinery they have, the majority of them is using hydrogen. Why? What does it mean? Well, hydrogen is produced and consumed so fast that never accumulates outside. The net production, the net accumulation in the environment is basically zero, because as soon as somebody is producing some hydrogen, somebody else is going to use it. And we see this in many, many, many ecosystems. Net accumulation of hydrogen, free hydrogen, is very, very low. Now, these are big implications, for example, in thinking about natural hydrogen in the environment. Because if microbes use it so fast, accumulating a lot of hydrogen becomes a problem if microbes are lost. Becomes a problem in underground hydrogen storage in certain cases, because if you're under the right condition, microbes may use a significant portion of it, and we'll see in a second. So remember, they're very diverse. This is another way to seeing it. Remember also that hydrogen is a very good electron donor, but depending on what you couple it with, the amount of energy, that's the delta G of your reaction, is very different. So at the bottom here, you see fermentation. These are the microbes that produce hydrogen from organic carbon. They make hydrogen, you see hydrogen coming out, and they get very little energy from doing this process. That's why fermentation works only when the majority of the other electron acceptors are gone. Otherwise, anybody doing anything else gets more energy, is faster, is going to overpower these guys, okay? So very deep in sediments and things, you may get hydrogen production. And then, if you couple it to CO2 to make methanogenesis, you get some energy. If you go to sulfate, you get more. Nitrate, you get more. Oxygen, you get the most energy out of it. As we were saying yesterday, the most powerful the oxidants you're using to reduce, to oxidize something, the more energy you get, okay? Now, this also tells you that if you go outside the marine sediments and you measure, you try to see who's doing what in different layers, you'll find exactly this line. So in the top sediment, people are using oxygen. When oxygen disappears, people use nitrate. When nitrate disappears, people use sulfate and iron. When this disappears, people use CO2 and they do methane. When that disappears, people ferment and hydrogen gets produced. Now, this is a ladder of utilization of oxidants that is dictated by thermodynamics. And it's very consistent out there in the environment. And you may think about your reservoir and think about all the electron acceptors you have in your reservoir and say, if I inject any oxygen, that's going to be used first and fastest. Why? Because it's the best oxidant. And people that can use it, we get a lot of energy out of it. If I introduce nitrate, nitrate is going to be used before sulfate. And in some places, this is used as a mitigation strategy, right? You don't want sulfate reducer to make H2S, you dump nitrate down. So nitrate reducers with ammonia, easier to remove, less dangerous. And sulfate reducers are outcompeted because they get less energy and they're slower. So you can use this as a strategy in thinking about microbiology. Now, there are microbial effects on underground hydrogen storage that have been described in a few pilot places. For example, in Kedzin, in the saline aquifer of Bayes, the sun storage, and the Lobodice gas town. In these places, people measured the production of methane, and they link this with the consumption of up to 10 to 20% of the injected hydrogen, up to, generally speaking, much less. Now, engineers, we call this methanogenesis, the making of methane by microbes. Engineers call it biomethanation, making methane biologically. Often is a desired product, like co-injection of CO2 and hydrogen underground. People wish all of that gets converted to natural gas, because then it's easier to compress it, liquefy it, ship it, use it, and so on. So there's a lot of research that goes into, let's try to figure out a way where we can sequester CO2 with hydrogen, make methane, then when we burn it, it's a net zero because the CO2 we can repump it down again. We know it's not going to be a real net zero because there are consumption, but let's assume it is for the sake of this discussion. We have not much evidence of H2 being used for many other things. So we have evidence for methanogenesis happening in some location, but we don't have not a lot of evidence for hydrogen disappearing in other ways. And so when you want to try to figure out how this disappears, you want to figure out how they can affect geological storage, right? And the first question you ask is, can they actually affect something as big as storage in a large reservoir? I mean, we're talking about pumping down million cubic meters of hydrogen and then the cap from very large porous media. What are the evidence that microbes can actually have an effect there? Well, first evidence comes from oil and gas storage, where microbiology can have a huge effect at a big study. The second study, which is closer to the things I do, comes from the effect that microbes can have on volatile cycling. Now, this is a subduction zone. You got a plate sinking under another one. And in the area, you have volcanism happening, okay? That's very common in Chile and Costa Rica, all the ring of fire around the world. It's also common in Italy with the Aeolian Island and so on. You got one oceanic plate sinking down a continent. What happens is that as the plate is going down, okay, the blue area is the theoretical space below 150. Porous rock, when you are in the area of, or fractured rock, and you are in an area where temperature allows life to exist, okay? And as the plate is going down, you're gonna get things that are released from the plate and coming back up. And they're going through the blue area, so they're crossing the domain of life and arriving to the surface. So a long time ago, we started asking, is it possible that microbes in that blue area are actually affecting the gases going through it? So this is not storage, this is flow through, right? We are looking at it. To me, when we started discussing this, it looked like a giant aquarium filter. When you have an aquarium, you have the volume of your water going through a porous media for microbes to remove things like ammonia, nitrate, break down organic carbon, and clean the water. Here we have a lot of things that are being released by the geological processes coming through porous media filled with microbes that potentially can affect it. So to test this, we went to Costa Rica. That's where we started this work. Now we've done it in many other places. So you can see here, this band here, what we call in oceanography the trench. That's where the oceanic plate is bending and going down the continent. And then you see portion here is called the forearc. And then the yellow names here are volcanoes. That's what we call the volcanic arc, which is related to the plate getting to a certain depth, triggering, melting, and getting a line of volcano. We have it in many places globally, very common. Now we started, every red dot you see there is sampling we did in aquifers that were coming to the surface, hot spring coming up. They're bringing fluids from depth up, okay? We don't know from how deep, but there are ways to discuss this later if you're interested. Now, when we went there, we measured microbiology, geochemistry, composition of the water, composition of the gases, we measured a lot of things. One of the things that was interesting is that when you compare the inorganic carbon, so the CO2 amount in the fluids, x-axis, against the isotope of carbon in the y-axis, you get this relationship. This is not a linear correlation, but it is a correlation, right? Things seem to be related to each other. When you have very little concentration, you have very light isotopes. When you have high concentration, you get heavier isotopes. Now, these are all waters. Those down there are volcanic gases, which should be what we call an end member. That should be the deep CO2 coming up from the volcano directly to the top, right? The interesting part is that this relationship was spatially distributed. Outer forearc, near the trench at sea, forearc, volcanoes going up. It's like, if you go back here, they move from here to there. So here, very little CO2, very light in isotope, then it gets heavier until you get to the volcanoes. So spatially distributed, right? Increasing distance from the onset of subduction, which for us was suggesting a process that moves, that is controlled by things going down, right? Because we follow step gradient. Now, it's actually not one line. If you see, it's already obvious, it's two lines. In geochemistry, when you see something like this, between a relationship like this between concentration and isotope, we call that a rhyolite distillation. It's a process by which a type of isotope, in this case the heavy one, is removed selectively by precipitation. You can model this trivial in geochemistry as calcite being precipitated. So CO2 was actually being removed by calcite precipitation. You can model it. You have the starting condition and you have the starting temperature. Look at the temperature at which calcite is modeled to precipitate here. 63 to 67 degrees. We are in the middle of the life temperatures, right? Smack in the middle. So there is a possibility that life might be involved in precipitating calcite, in removing CO2 from the waters. So the idea here is that while the fluid are coming down, they're reaching CO2 from the bottom, from the recycling of the plates and so on. As they go through the porous media, CO2 gets removed. And as it gets removed, there's calcite precipitating. And there's a lot more precipitation here, in the outer forearc and forearc, and very little up here. And we can see this, we can model this, and then the next question becomes, okay, is calcite precipitated? But are microbes involved in this, right? Because this is now CO2 going to calcite. There's no direct microbial involvement. One other thing we've noticed is that the inorganic carbon, you see at the bottom, is related to the organic carbon. And actually, the delta of the isotopes between one and the other changes always cost to volcano, but it's pretty much constant between the two. In other words, every time you measure CO2, the organic carbon, the sugar present in the water, they have a similar offset in isotope space. Now, depending on the direction of that offset, biologists are able to predict what metabolism is present. The offset was very constant throughout. You need to do just pairwise in every site, pretty constant, and that suggests that microbes are producing organics, glucose, lipids, everything, of the CO2 that is coming from that. That suggests that microbiology is removing a lot of CO2. And if you do all the calculation together, it turns out that 40% of the CO2 that our calculations originally, volcanologists, thought it was going down into the mantle, sequestered forever into the mantle, is locked by this porous filter by biology and calcite precipitation. So our calculation of where CO2 was going was off by 40%. That's not a small number. 40% is a big number. And telling that in a place as big as Costa Rica and Panama, 40% of the CO2 of the entire area is being removed by microbes in the crust is a big claim, right? Now imagine if you can expand that to every subduction zone globally, every place. Imagine if you can make similar claim for the amount of CO2 that you're pumping down in carbon sequestration experiment and saying 40% of what we pump down is used by microbes and locked in place doing something else. That's fantastic. That's everybody's dream. On the contrary, if you're pumping hydrogen out there, it turns out the microbes are using 40% of the hydrogen you're pumping down to do something else. It becomes a big problem. So we have evidence that microbes can actually interact a lot with these big scales, and we need to figure out why and how, especially for hydrogen. We repeated experiments like this in a place that is very different from Costa Rica, which is Southern America. What's the big difference there? Both of them have a plate going down. But Costa Rica is a very small, tiny country, has a very thin plate. When you go to South America and the Pacific plate is going down, you create the Andes mountain and plateau. That's one of the thickest crusts globally. It gets down to 80 kilometers of thickness. And we know microbes can go as deep as temperature gradients allow, right? So we did the same calculation and experiments. This is a bit more technical, but what we saw is that when you are in the central area of South America, CBZ, where the crust is very thick, we see a lot of what we call crustal assimilation, addition of CO2 in the area. When we move to the south part of South America, where things get thinner again, we see a lot of calcite and microbial precipitation again. So right now we think that microbial precipitation and calcite precipitation is happening everywhere, but you don't see it sometimes because you see more input from the crust. In other words, when the crust is thin, microbial calcite, that's the theoretical depth at 150, the relative contribution of these two is more similar. So you see a microbial effect. When you have a very thick crust, microbes don't really go very deep because gradients stop them, but you have a big effect of the crust. So it gets overpowered. Now, thinking about what this graph is about, is again thinking about biotic to abiotic effect of what we're seeing, right? And the trade-off between the two. In this case, is microbial processes, calcite precipitation and incorporation into biomass against crustal effects, effect of a very thick crust. And the relative contribution of the two is going to tell you what things look like and what you should think of. So yesterday we were talking about rates of two processes. In this case, we're talking about magnitude of two processes overprinting each other, which is going to be the case every time you do stuff underground, right? You're going to have geochemical effect, geological effect, and biological effect overlap. Now we do a small break, and if you have questions, we can do questions. Otherwise, we do a 5-minute break.
Course, it would be a great step versus increasing.
Yeah.
I don't know which is the velocity of the calcite and microbial sink.
We don't know.
Because the crustal assimilation we know is pretty low.
We think it's pretty low also the top one, but we don't know.
Okay.
We can assume steady state of the cell and assume a certain turnover time, but that's a lot of assumption without direct measurements. Yes.
During this calcite presentation equation, that overall gas decrease is decreased that is converted to the solid phase as calcite.
Yeah.
And in the red study, I didn't think I just noticed in a geological scale, like in a field application, I would assume the pressure in the reservoir is that your pressure should decrease. Like
Yeah, you're thinking about a closed system in a reservoir with cap rock. In this case, we were in an open system. So pressure doesn't really change in an open system if you have flow through. But in a reservoir, you're absolutely correct. As you move things from gas phase to solid phase, even if you move H2 to reducing H2S and then H2S reacts with iron and makes pyrite, you should be removing from the gas phase. And in this case, you should reduce pressure inside.
yes, like now there's a trend like storing CO2 to the balance, like to convert it to like
Calcite.
calcite. I don't know. I don't think there's like a pilot study that shows those kind of pressure drop. Like
No.
It might
Only
be
as far as I know, only theoretical, right?
All right, a lot of experiment proposal that big
also
scale.
the reaction rate is that could be smaller than
Very low. Wow, the-
I was thinking that geometry is not as not a big one.
I think I think you showed me a proposal where people wanted to put some basalt or, I don't know, periodic rocks in somewhere in the coastal, and then waited to see what the CO2 was doing,
Yeah, they wanted to grind up and dump ultramafic rocks at shallow depths in the ocean, and then wait for natural carbonation to come from CO2 drawdown from the ocean.
And they were assuming the reaction was fast.
Well, but that's a big problem right now in serpentinization, right? We don't know in reality how fast is the reaction at low temperature and pressures. Some people claim it's very fast, some people claim that it's impossible, and we are still nowhere to a definitive response.
Talking about the CO2 injection on the salt, have there been any microbial investigation on that? Like, what is?
Yeah, that's a few paper, more than a few paper that look at microbial composition in carbon capture storage pilot sites. And in some places they demonstrated very good methylation, so very good production of methane. And there's a big debate to understand where the hydrogen is coming from in these sites. Because you pump down CO2, right? You don't pump out hydrogen. So hydrogen might come from a different variety of things. Microbial fermentation of organic carbon, radiolysis of water, thermogenesis of hydrogen from deeply seated organic rich layer that comes up. You know, we can serpentinization or oxidation of iron-rich rock lenses somewhere that we don't know of. But at the end, if we see methane coming up from these sites, we know it's microbial and we know there's hydrogen. We start just debating where the hydrogen is coming from in that case.
But how about the process of CO2 consumption?
Well,
Like the one.
as far as I know, nobody ever looked for possible calcite precipitation in current carbon storage things. I'm not aware of that. You need to think that carbon storage so far has been done in porous media or salt cover.
The moment, surface, yes.
We are talking about CO2 moving through volcanic igneous rocks. So here you have a lot more potential for cross reaction with iron and processes like carbonation linked to serpentinization. When you are in porous media, you're far away from the condition where you can have active carbonation linked to serpentinization. So the process we see here,
Unless you use carbon.
unless you use carbon. Yeah, but even that you need to be careful because you're pumping CO2 and that affects pH and might affect carbonate dissolution. And microbes that ferment, the microbes that ferment if there's organic carbon around, produce weak organic acids that can dissolve carbonates. So there's a lot of things that might go in different directions there, depending on where you are. Plus carbonate may contain sulfate to an extent.
Interesting. However, I think that the sun experiment that you were on the right, one of my advisors of the SHINE is the president of the. So
We can ask him.
we can ask him if he can send some rep, because I have sometimes I look at their website, they're not updating much.
Yeah, we can ask if they have any evidence for
No, but then
calcite
I'm
precipitation.
referring to the projects in Iceland where they direct inject CO2 to the salts. And they're claiming like in 2 years you can sequestrate that CO2. Like I don't know how many
I don't know.
percent or whatever, but I was just wondering if you can do some microbial testing.
You know, Iceland is a pretty hot place when you start to go underground. So it's not a great place to go deep with microbes. Heat flow are very high in the area. That's why they can do a lot of geothermal energy, right? Because they have very high heat flow. They can use high enthalpy geothermal energy. 5-minute break.
So
I would say, should we ask him when we're all back?
Okay.
Thanks.
Because we projects that.
I think a month ago, some of the resources from that project presented something that that's how I know.
Yeah, that's the thing, just very looking at it, we know the. If I get it.
Andate a fare il carico di pelle, oltre a quello che c'è fatto il break normale, per paura di rimanere senza. Ora,
Un attimo.
arrivo. One, two, three, 4, 5, 6, 7, 8, 9, 10. What do you mean extra? You're extra. Okay, so we're missing one. Cominciamo? Still extra.
Everyone is there.
So let's move to the actual hydrogen things, okay? So this is a schematic and idealized and simple cross-section of processes that might generate natural hydrogen. You see radiolysis, serpentinization, you see biogenic, thermogenic hydrogen, and as they relate to natural hydrogen spring or hydrogen traps found in the geosphere. And then you see the white hydrogen, which means just exploiting this reservoir, green hydrogen, you know very well, coming from pumping down and storing hydrogen produced through renewable energy sources, and then orange hydrogen, the stimulation of the oxidation of iron, oxidation and production of hydrogen, either through iron-rich rocks or, ultramafic rocks and things like that. And then we have the magmatic hydrothermal hydrogen, which is one way our planet makes hydrogen, but we think that's very little relevance for the usage, because it's hydrogen that is produced in high temperature atmosphere generally gets lost very, very quickly around. Now, one key point you should keep in mind is that many of the problems that relate to green hydrogen, orange hydrogen, they also relate to white hydrogen, especially in thinking about cap rocks, type of place where it can accumulate, activity of the microbes that might consume it. And so tomorrow morning, you spend the morning discussing only methods to analyze microbes and their activity and microbial diversity. So we'll discuss some of the things we've been mentioning earlier through your questions. And then in the afternoon, they will show you what we are doing for underground hydrogen storage and natural hydrogen and what type of experiment we've been doing. Essentially, though, when you want to interact with the microbes that are present in places like this, like in porous reservoir and salt cavern or hydrogen-rich sites somewhere, what you do is either well sampling, so you try to sample formation waters from depth and try to analyze indirectly most of the time what the community is composed of, what are the geochemical characteristics of the water, what signal you have that might suggest that microbes are or are capable of using hydrogen at depth. Or another popular type of experiment is what we call microcosm. Now, micro, meso, megacosm depends on the size of your vessel. But microcosm usually use volumes that go from 100 ml to 1 to 2 liters in closed bottles, gas-tight bottles, where you can put formation waters or waters you sample from different locations. You can include solids like cutlings from the drilling, actual sandstone or mudstone or shales or whatever you're using, and a gas space where you can then, those are overpressured. You can tell from the top, rubber top. You can put either the gas that comes from the reservoir or excess gas of other types. Now, in this type of incubation, what you are looking for is changes in chemistry, gas composition, or microbial composition over time. And so people sample this, keep sampling every day, every week, every month, or every few months, or every hour, depending on the speed at which you expect things to change. And then you may or may not do some addition. Addition meaning you may add excess of something. You may add excess hydrogen. You may add excess CO2 because it's going to be your cushion gas. You may add oxygen because you think you're going to contaminate with oxygen as you work on your reservoir. You can simulate things around. Now, this approach is the most popular approach right now to study how communities respond or are responding to underground storage. And if you go through the literature of the studies that I've done, you see things like this one. So this is a very recent paper of Frontiers in Microbiology. What you're seeing here is two experiments, top and bottom, in which they have added H2 and CO2 in the gas space, and then they added two organic substrate, lactate and acetate. And now so top is acetate, bottom is lactate as an organic carbon, and then you can see cell numbers, sulfate concentration, acetate concentration, lactate concentration, and then again cell number, sulfate, acetate, and lactate. The three lines, light to dark, are three different concentrations of salt. So we go from 2.6 molar NaCl to 4.4 molar NaCl. Now, if you just focus on the top left, you see that microbes grow over time in these experiments. Those are days, 3 months of experiment, 120 days. You can see microbes grow over time. They increase in abundance. So they are happy, they're growing. And they grow faster when salt is lower, less stress on the community, but they grow in all three of them. And you can see sulfate goes down very fast in the low salt concentration, but it doesn't get used in the high salt concentration. At the same time, you see acetate that is going up, lactate is basically not produced, but we know was also not added from the top, right? In the bottom, where we added lactate, you see microbes going up faster than the other, sulfate being used in the lower concentration salt, not used here, acetate goes up as lactate goes down. What is this telling us? Microbes are growing, they're using hydrogen, most probably. There is sulfate reduction going on, only a low level of salt. So people use in this paper this data to conclude that, well, if we are in a highly saline system, sulfate reduction might not be a problem. So don't worry about H2S being created, because at high salinity, sulfate reducers are not working. Now, this might be a bias for the inocula, the original brine you used for this one, right? In your original brine, there were no sulfate reducers. But this is not the case, because they're going down at low salinity. So maybe there were no sulfate reducers adapted to very high salinity. It might take very long for somebody to evolve that can actually do that at high salinity. We don't know, only 120 days. But this type of experiment, they become the de facto what microbiologists do. One key point here is that you are stimulating the community in most cases. You are providing acetate and lactate, which is not necessarily present in your reservoir. But why we decide to use acetate and lactate? Why those compounds? Why we're not giving pure sugar, which is a very good substrate for all life? If you remember yesterday, we discussed fermentation. Fermentation is a pathway by which organisms take organic carbon, sugar, and then they break it down without an electron acceptor. So they start to break it, and then they dump electron on itself, and they produce lactate, acetate, ethanol, and another organic acid. Now, glucose up there is an intermediate of other metabolisms. You get to glucose, for example, when you start to break down hydrocarbons. So you take hydrocarbons, break down, catabolism, break things down, and then every cell needs glucose. So you use the energy of breaking down the hydrocarbon to make glucose inside your cell. And then if you don't have an electron acceptor or somebody else eats you, the glucose might go to acids, organic acid, lactate, acetate, propionate, butyrate, and a number of other things. So people are saying, we'll add to our experiment lactate and acetate because I expect fermenters to be there, and I expect them to produce this compound from any organic matter that might be there, including dead other cells. One thing to consider is that when you are in a complex population, microbes do not only compete for resources, like multiple microbes might compete for hydrogen to do different things, but they also exchange resources. Hydrogen can be one of the resource exchanges between cells, but are also directly affecting each other. If you look on the other side, that's a list of possible interaction. There are microbes that are predator of other microbes. There are microbes that will eat microbes that die in the population. So you need to think that there are cells that are made and broken continuously there. And that's organic carbon. A dead body is organic carbon. A dead cell is organic carbon. So you may have very complex dynamics. And that's something that we see in experiments like this sometimes. When people run this experiment for a very, very long time, they see suddenly fluctuation in population. Yes. Yeah, probably it might be inspired at the expense of sulfate. Lactate is a very good substance that heterotrophic sulfate reducers use in sulfate reduction. So they destroy the lactate at the expenses of sulfate. And or lactate may be used by some bacteria to produce acetate. Acetate is shooting up. They didn't add any acetate, but acetate being produced. Acetate is a great substrate for microbes that do methane from acetate, acetoclastic methanogens. So you are kicking off a series of side reactions just by using or consuming some of this. There's an experiment started many, many years ago, maybe 25 to 30 years ago, which is called GASP, Growth Adaptation in Long-Term Stationary Phase, GASP. So what this guy did, when we grow microbe, and we inoculate a microbe in a new environment, there is rich of food and things, microbes start to grow, they go up, and then eventually they flat out when they run out of space of food, because we are in a closed system, right? We are in something like this, they run out of space of food. And typically, when you go up to the sigmoid curve at the top, things start to die off, and people will get it and make it in a new virus with new media, new space, and the microbe grows again. We call that microbial growth curve is a sigmoid curve. Somebody said, okay, what if I wait longer than normal? And they saw that microbes will go up, flat out, and they start to die. People will assume that that's the end of the things, right? They die, they don't have food. Instead, somebody kept that culture going for months. Grows in one day, keep it going for months. And they kept monitoring the growth. And they saw that things will go up and then start to oscillate. Up, down, and then have oscillation. So they will never die off completely. And there are actually moments in which population goes up again. And what they figured out is that within a single population, they start to appear, to evolve, like we see in the experiment of yesterday, subpopulations. And some of them die off, another population eats the leftover, and they grow until they die off, and somebody is better at eating their leftover, and you have complex population dynamic that oscillate over time. And you can keep the experiment going for 3 to 4 to 5 years, and you keep things going up and down with complex dynamics that are very difficult to predict. That's exactly what's going on in natural population. Things are not stable, always same composition. You have continuous changes in relative concentration. We don't see them sometimes because we stop early. Now there's a new paper from my colleague Carol Lloyd, the same person that wrote The Time as a Microbial Resources, where they did H2, CO2, methanogenesis, and sulfate reduction in a closed vessel for 3 years and a half. And there are very interesting dynamics that kick off after a year and a half to 2 years into the incubation that you don't see earlier than that. Because conditions, they just need to mature and get more reducing, things need to go in the right direction.
With temperature and pressure that they manage to reduce in there.
This one?
Okay, no.
Oh, Karen Lloyd is 1.2 bar and 30 degrees or 35 degrees.
Okay, so soil.
Yeah, marine sediments.
Marine sediment.
Marine sediments. Many of these experiments done like this, they're actually done around 2.5 bars, maximum 3 bars, which is the overpressure you can put in the head space of this type of vessels. So they are somehow related to what happens at that. Now, we have some papers published by people working at high pressure that say when you have pure hydrogen at high pressure, so 30 bars up, the solubility of hydrogen is dependent on your pressure, right? On the partial pressure of the gas. So if you're doing an experiment with 100% hydrogen, but you are at 1 bar, the actual absolute amount of hydrogen you dissolve in the brine is small. If you start to pump the pressure up, you dissolve a lot more hydrogen. And it seems like this experiment suggests that microbial activity is inhibited when you are at very high pressure with a lot of hydrogen. So it looks like it might be actually beneficial to be a very high working pressure for your reservoir rather than low working pressure. Now, these are experiments run in 1 ml volumes in microfluidics. So scaling things up and thinking is different. But at least we see microbes that if you lower the pressure, they use the hydrogen, increase the pressure, they stop. This is work by French colleagues in Bordeaux. Very, very interesting work. Yes?
What is that?
we think it's a problem of affinity of the enzymes. In most cases, enzymes are used to work at either low pressures with hydrogen produced like in geothermal system, or high pressure but very low hydrogen concentrations like in deep sea hydrothermal vents, where there is hydrothermal hydrogen produced in deep sea hydrothermal vent, but we are in the nanomolar range. We are not 80% of the gas space, right? And you might just be poisoning the enzymes, too much coming in too fast, and the enzyme cannot ever release substrate. And so there's no activity, but this is hypothesis at the moment.
All those like microbes or enzymes are affected cyclic pressure change.
Well, that's a really good question. There's an entire branch of biology called biophysics, where people have been dealing with conformational and functional change due to high pressure. Now, at the moment, there are only 5 or 6 laboratories in the world that are looking at adaptation of enzymes and microbes to high pressure. The problem is that we are doing it with a few model organisms we can grow in the lab. So whatever we find then, we think might be accurate with the environmental microbes, but we don't know. So another way to tell you is that we don't know. That's the short answer. So addition of things is very popular, but that gives you an end member, right? We adding something that might be there, but we don't know. We are hyper-stimulating the community to respond because we are pumping them with acetate, we are pumping them with lactate, we are giving them sulfate. So these are all ideal situations we are testing in the lab that are not necessarily representative of the real activity under them, right? And something instead, the wells, to compare with the response of the community, might tell us something more. So now many laboratories, including my own, are coupling laboratory experiments with well sampling and try to figure things out. And one thing that Martina will speak tomorrow is that we started using, we put in several proposals, natural hydrogen spray as a good analog to explore the state space. Because, you know, there's only so many wells you can access, and it's difficult and it's expensive. But if you want to see hydrogen activity at every possible pH from 1 to 12, you don't go in wells. You use natural sprays, and then you can cover the entire pH range. You can cover the entire salinity range. You can cover the entire temperature range and start to see what microbes are using hydrogen where, what they couple it to. Is it true that they can couple hydrogen to sulfate at every temperature, every salinity, every pH? Or there are a space in this combination that is used and a space that is not, that we don't know why. And we're trying to explore that also from the genomic level, and that's Guillermo work, because we have genomes sequenced of thousands of microbes, okay? So we are going into genomes and looking, do they have hydrogenases to use hydrogen? Yes, or which type? And then what type of electron acceptor can they use? And we are linking the two to see how many times this co-occurrence happens all over the place, or if it's rare present only in a certain group. So it's true that that type of hydrogenases is always present when sulfate can be used. Or maybe there are multiple types of hydrogenases that are present when sulfate is used. We have 17,000 genomes right now, or close to that. Each one of these codes for multiple hydrogenases at terminal reductases, the enzyme they use terminal electron acceptor. So Guillermo is building the pair up to start to see if we can learn something about the coupling of these two. Because at the end of the day, the other things that we are doing that I think that should be done is you have your reservoir, that's worked by Martina, Guillermo, Gabriele, and other people in the lab. You have a reservoir, you write down the reactions. Now, these are not all the reactions. Martina wrote down 74, 75 redox reactions that are possible with hydrogen in a reservoir. And we limited ourselves to very few minerals, because if you start to add minerals, your state space, combinatorial space explodes, right? And then you do this, and then you start to think how they react to each other, how do they interact, right? You see ammonia there, and then ammonia can be used somewhere else, and then you see elemental sulfur here, but elemental sulfur is also the product of sulfate, and then things might cross react. The reality what you have is not a single equation, it's a network of reaction, right? Or something like this. Hydrogen is at the center, because that's what we're looking at. Every number there is one of the reactions you see on the right, but there are a lot of cross reactions that can happen in the background. That we have them mapped down, we're just showing a few of them for simplicity. But the idea is, can we model this? We were discussing this yesterday, right? Can we model this abiotically? And can we see how does it, what's the distance with the real life? And can we actually then understand where microbes are working based on the results? Reality versus modeling. Reactive, redox network reaction modeling versus geochemical measurements in pool and reservoir. So what does it mean for you guys? You're not microbiologists. Well, Guillermo is, but you guys are not microbiologists. What does it mean? Well, I would approach things like this, and I often show this to my geological colleagues when we interact and discuss. So ask yourself, in my process, whatever process, or part of my process, or path that my proxy is taking, for example, you are sampling something on the surface that is leaking out, so it's walking through something, right? It's leaking through porous media or going to water. So is my process part of my process or part of my proxy in the boundary domain of life? So -13 to plus 135, 0 to 12.8 pH, below boiling, 0 to 35% salinity, 0 to 2 GPa, and pore space. We should add to that list. So if the answer is no, geology rocks, you're good to go. You don't care. If your process, part of your process, or any proxy you're using is passing through that domain or is present in that domain, then the answer is yes. So now you need to ask a new question. Let's go back for a second. Look at this figure I showed you here. There's a line there, which is arbitrary line we put down, which is the 122 to 135 isotherm. If your system is above it, worry. Your system is below it, don't worry too much, right? That's exactly what I'm telling you. So you ask yourself this question, no, good to go, yes. The next question should be, how much time and space has life to interact with my process? We were discussing yesterday, if you're studying underground for 6 months, you probably shouldn't worry too much, right? We're not sure, but probably it's not a lot. If you are studying for 20 years, then life has a lot of time to adapt and interact with yourself. So ask yourself, how much time space has life to interact with it? For example, in very thick porous crust, where fluids are coming up very, very slow. So one thing, as I was telling you, one very important parameter is heat flow. So let's consider this for a second. We have temperature, pH, heat flow. Forget the volatile origin, that's more for people working in other systems. And then depth of your subsurface biosphere on the right. That goes from centimeters surface to very deep, kilometers. Now, depending on where you are sitting in a geological settings, heat flow changes, right? If you are in a volcano, closed vent volcano, like Campi Flegrei up here, the heat flow is going to be very, very high in the area because you have shallow heat sources, right? Well-developed geothermal system, which means that generally temperature of anything you measure at the surface is going to be high. pH will be generally acidic, which means the depth of your subsurface biosphere will be very shallow. Why? Because as you go deep, 1 meter, 2 meters, you immediately found boiling condition. Contrary, if you are in a deep fault system, where your heat flow is generally low, can be very variable, look at HF, very variable, but can be also very low. Temperature, they can oscillate depending on where you are and how fast fluids are coming out from them. pH can be variable, which means that your subsurface biosphere can be from shallow to ultra deep, depending on the settings where you are. Now, if you can ask yourself this question regarding your reservoir, where is my reservoir sitting compared to where the theoretical boundary for life is, then you're able to answer the question. We did this exercise for a number of settings to help people classify and think about how deep the subsurface is. We need to finish this paper and submit it soon. So how much time, space has life to interact with it? Little. Things are very shallow. Things are too hot. Things are too fast. Things are coming up very fast in natural system. Things are bubbling very fast, very up. And there are geochemical ways you can understand all that. Again, geology rocks. You're good to go. No worries. But if the answer is a lot of time or space, the next question becomes, okay, Biology might be doing it. What's the relative contribution of biotic to abiotic processes? We go back to this, right? So we are in the boundary domain of life. There is time for life to interact with the process. How much is it going to interact under my condition? Should I worry about the abiotic reaction of hydrogen with the rocks? Or should I worry about the biological interaction of life, hydrogen, and rocks? Which one of the two? So the next question becomes for you, are the rate known of biotic versus abiotic? Great, you can calculate and figure out what's the contribution. But bad news for you, in the majority of the cases, we don't know the rates. Unknown. So what you do? Squishy things are messing up your work, call a biologist. So in essence, that's would be how, if I was a geologist, I will approach this. We know we have a variable, we have a process that might affect a lot of the things you interact, from the porosity to the rock mechanics, to the contact angle of the gas with the rocks and the fluids, to the surface tension around brine, because microbes can make surfactants and things that lower surface tension, which is going to increase water wettability and water exchange. They can mess with a lot of these things. So ask yourself, am I in the domain of life? Is there time to interact? Do I know how they're interacting? If the answer is no, then you need a microbiologist on the team. If you're outside this, you're good to go. No worries. Now, with this one, that was the last slide, which I think is how to. At the end of this, and now tomorrow we're going to discuss a lot of practicals. We do an exercise together, we build something together, we see what we've been doing as a lab. But essentially, the theoretical portion of this course is over. So at the end of the day, there's a lot of microbes, they're small, they can interact with all the redox chemistry that might be going on naturally out there. The how much is what we need to figure out. And by the messing with a lot of things related to your work. We didn't went into detail, but if you look, microbial effect on wettability, microbial effect on porous structure, microbial effect on solubility of gases, you'll find hundreds of studies that they tested this in the environment, in laboratory condition, under simulated pore condition and so on. So what I want you to take back home from this is just being able to ask this set of questions and have the sensibility to think that some things you are measuring might have been altered by interaction between geo and bio, which is what, you know, we have this big division between geology and biology, but in the environment, things don't care when you put the boundary, right? Things are happening in the same time, same space, together. Now, questions?
Right. Let's start. Just the last sentence you say, because one of my okay, let's put ourselves in the context where most of the reservoirs stay within the boundary that you are deciding to find there. Most of the reservoirs we are looking is stay between 0 and 155 degrees, which depends which is the geothermal gradient. In some cases, like the classical basin, what we call offshore basin in a rifted system, or in the continental system, in the first 5-6 kilometers, that means almost on the place where the company wants to inject stuff, not too deep, not deeper than that, because it's too expensive, and therefore you have to deal with it. My understanding is that for the real, what makes the difference, once we have sampled, mapped, see what's happening, which kind of community, microbial community is there, is to understand which is what we call the time space, basically the space dimension of those kind of interaction and the timing, which what is obsessing because not very clear how much of these tones of reaction may really affect the system. My other question is that one. If you look at a lot of reservoir basically works done by essentially engineering versus essentially geology, looking at the microstructure of the pore pair. In the subsurface, all the rocks, all the porous media, the rocks are saturated by a fluid. Can be single phase if you're lucky, brine, can be brine methane, can be brine methane CO2, can be brine petroleum CO2, etc. And then the important part at the core scale is to understand what's happening in terms of wettability of the boundary wall of the porous medium. Now, in the last sentence you say is that you just say that there are a lot of studies of how microbial systems interact at that scale in this kind of system. Can you tell us a little bit more?
So there is work that has been done, for example, a lot at the interface between brine and petroleum and the communities that are present there. And we know microbes are very good at producing surfactants that increase the interaction between water and petroleum at the interface. So they increase the mixing, essentially. That creates micro droplets of oil dissolved in water that in natural conditions are not present. And microbes increase that exchange. There are studies that show that if you get grain size of the same type, but they have different smooths. So one is very smooth, one is very coarse in angles. The growth of microbial biofilm on the surface, they can change the incidence angle by which the grain interacts with the fluids. And depending on changes in viscosity of the fluids, that changes the wettability a lot. So we have a series of studies on this that point to different directions. The problem is that many of these studies, they're specific to one type of microbes. And sometimes when you change type of microbes, you get contrasting evidence. Because one microbes may produce a lot of surfactants, the other one no, produce exopolysaccharides. And so the effect on wettability changes completely. So we know they can affect this. We're not able to predict it because we don't know how complex communities will behave under in situ conditions.
Because on the hydrogen case, I mean, one of the biggest questions, but several groups, I mean, the Hardy groups and also I think the Cation has been started, has been working a lot on this kind of.
One of the papers is from Hardy and Nicole Doppler on the wettability of with and without biofilms, for example.
Right. My question is that, I mean, in those experiments, how fast is the, I mean, how much impacting is the process of those microbial system respect to the, I mean, solve the equation without the microbes and solve the, let's say, equation of the reaction, understanding all the wettability is distributed, and therefore, how the hydrogen react with the microbial.
Yeah, but the point is that these experiments are rarely more than 3 months long. 6, the longest we have. Why? Because they are the most time a PhD student can dedicate to an experiment. We are essentially limited in time, but no, you're laughing, but that's the truth. If I wanna run a 5 years experiment, but the average PhD is 3 years to 4 maximum, and they need to do other things, including classes, write, and so on. And the average project we get financed is 2 to 3 years. I need to connect multiple projects to run an experiment for 5 years with multiple people, and that's non-trivial. And so the majority of the experiments, they run for much shorter than that. And so whatever results we have is bound by the boundary condition of experimental settings that are generally short. So if we do experiments that are 3 months, and our injection time in the reservoir is 3 months, we are well simulating the system. But if we plan to inject and keep stuff down there, at least partially, because we're going to produce and pump, but there's going to be a block of hydrogen there for 10 years in a row, a 3 months experiment is not going to tell us anything. You made me think about one thing I want to clarify. The boundary conditions there are for life, all of them, right? But for example, there are limits of specific life. Photosynthesis, the production of energy, extraction of energy from photons, does not exist above 70 degrees. Non-existent. We know 0 microbes that can do photo anything above 70. We think that's due to the stability of the chemical links that are needed to capture the photons and so on. So if you ask yourself, no, what's the boundary of life, but what's the boundary of a specific metabolism? Many people right now in underground hydrogen storage are asking themselves, what's the limit for sulfate reduction? Because they're interested in H2S. So they're asking that. And there are papers that suggest that above a certain salinity and above 75 degrees, there's no sulfate reduction. Now, that's very good to know for the microbes we can grow in the lab. These have not been tested in the environment. That's due to microbes we can grow. And we know we can grow a very small fraction of microbes, right? So it's a partial view. The other things to consider is that H2S is not produced only by sulfate reduction, but also sulfide reduction, also thiosulfate disproportionation, also elemental sulfur reduction. That's a lot of other microbes. We need to figure out at what temperature they can do it. So sometimes it's not relevant just to ask what's the limit of all life, but what's the limit of the metabolism you want or you don't want in your system. Now, that might be a lot more test, but might give you a window where some of the reservoir are, where what you don't want is not there. And that's what many people are chasing right now. No absolute absence of life, but conditions that are not favorable for certain metabolism. Sulfate reduction is easy, right? Just pick a reservoir where there's no sulfate. Easy, up to a point, because they can use other things, right? But that's the general thinking right now out there. Other questions? Yes.
Majority of the different gases or underground the temperature starts from 72 up to maybe 400. It depends about how deep they are. And this domain of life, if I remember correctly, it started with 7 Celsius and it increased now it's 122.
Well, that's the limit. That's the limit of life. Yes, in the 70s we thought that was 70 degrees, and then we've been increasing it.
It's not like the in situ conditions for the microbes, but you push them to live or to adapt and to live that conditions.
No, in this case, this is where microbes are perfectly happy. Actually, if you bring them a lower temperature, they die. So we are not, the microbe that we grow at 122 at the lab, we didn't push it at that condition. It grows better at that condition than it grows at lower temperature and pressure. It's a microbe that is usually found at deep sea hydrothermal vents, at 300 bar of pressure and hundreds of degrees of hot seawater coming out. So when we discussing subsurface microbes, those microbes, they're there and they're happy to be there within that range. So we will change the condition on them by pumping 100%, 80%, 90% hydrogen. But everything else is going to be where they grew and they adapted their entire, not just life, evolutionary history. So they're perfectly happy to be there. So sometimes we push microbes to adapt, but in many cases they're perfectly happy to be there. Other questions? No? Okay. Well.
Can you go back to some of the slide?
Which one, David?
Here you go.
What?
Next one.
This one.
Yeah. So those series of basically equation, I would say, of reaction.
Redox.
Balance
redox reaction. That's all there.
And they have been collected, I imagine, by all sorts of literature without looking at the context.
Yes, it's everything hydrogen can react. That's the theoretical space of hydrogen reactivity, without considering minerals, otherwise it gets out of hand. We just considered a few minerals in Martina's review. So the point is that...
Because my question was, okay,
how likely they are in underground reservoir? Some of them very little. Look at the top one. Hydrogen peroxide oxygen gives you water. That's called null gas reaction. It's basically the same explosive reaction we can use for rocket fuel. Some microbes live off it. How likely it is that we have hydrogen, oxygen underground with hydrogen? Very little.
The old sulfur and pyrite and all the iron, whatever, and relative iron related. I mean, probably all the reservoir got some of them, but they have some places are very minor.
But that's the thing, we need to figure out. So every one of these single reaction has a delta G associated, which will change depending on what reservoir we are thinking, because pressure, temperature, pH, salinity condition change, and ionic strength changes, right? So every one of these reactions is going to be more or less energetic depending on the reservoir. Now, on top of that, many of these reactions will be limited by the availability of products or reactants. So you may have a reaction that is still very energetic under reservoir condition, the top one, for example, but at the same time, if your reservoir oxygen is picomolar to 0, that energy has a density for kilogram of rock. It's very, very low because it's going to consume the little oxygen available immediately. It's going to be gone. So might be energetic, might be very fast, but the energy density for kilogram of your rock reservoir or brine or whatever is going to be very, very low because concentration of reactants are very, very low. Therefore, its significance in the context of that reservoir becomes essentially 0. The point is that if, in my opinion, and this is my opinion, if we want to approach without bias what microbes can do underground with hydrogen, we need to start from the biggest possible theoretical sets of reaction, model it for the reservoir we are discussing, abiotically, and then constrain it with real measurement from the reservoir. And then we might be able to predict overlaying gene information. So, oh, we're predicting the sulfate producer. Do we see them in the microbial community? Or do we not?
Yeah, that was my actually question, you answered it. So when you write down all the reactions, you actually go to that specific reservoir, take the samples, and you cross-check the existing knowledge.
Yeah.
With the reaction that are possible.
Yes, we cross-check the existing geochemistry and minerals and microbes with the reaction. Do we see anybody that can do reaction 1? No, we don't in the community. And plus it out. There's no one that can do it. We know oxygen is limiting and not there most of the time. So even if it's energetic, it could be fast. Its relevance for this reservoir is probably 0. And which means that in that case, it's producing water, who cares? But in other cases, that reaction might give something that then is used down the line. For example, thiosulfate here, sulfate reduction to thiosulfate. Thiosulfate is then used to produce H2S. But if we don't see anybody with the molecular machinery to make thiosulfate, and we don't measure thiosulfate as an intermediate in our reservoir, so we can think those two reactions by cascading one to the other, they're probably not very important, right? But this is an exercise that we just started doing in this way. So this is what I think is the right approach to get an unsupervised approach to the system. Otherwise, we constantly say, oh, we deeply care about reaction number 8. And everybody's just looking at reaction number 8. Because that's classic hydrogen-driven sulfate reduction to sulfide. That's part of it. That's one of the reactions that's very important. But we are not looking at cross-reaction. Which, you need to approach this as a complex system. You know, we always need to simplify to get first principle. But sooner or later, we need to face the reality of a complex system. And so, my idea, and what I'm really pushing also in the new project we have on hydrogen storage, European project, which I've been pushing with the PhD of the people working with me, is to do more of these larger overview networking exercise, like redox reaction network, and then try to see how that compares with the real life. Otherwise, we keep focusing on a single reaction and say, I added sulfate, it's doing it. No, sulfate is not doing it. And we're going to conclude from that in the entire complex community where there are hydrocarbons, minerals, different gases, different microbes, different rock composition depending on the layers where we're sitting, fractures and things like that is behaving. We're simplifying the system a little too much at that point, which is a needed step at the start, but we need to move into more complex things.
But then the extent of that damage or effects to the reservoir, to the hydrogen content that we're storing is another factor that we have to consider, right? Like if we're going through all these processes and then in the end it's like 0.1%.
Who cares, right?
Who cares?
But the point is that we don't know,
Yeah.
right? I wish microbe will be completely inconsequential to hydrogen storage. Because you already have enough problem with the reservoir integrity, caprock integrity, stress on the rock type, wettability, recovery, and everything else. If we can just say confidently, pump hydrogen wherever you want, don't care about the microns, that's one less problem to be solved, right? But at the moment, it's an open question, like many other open questions. So our role is not to demonstrate the microbes are important or demonstrate the microbes are not. Our problem is to answer the question, whatever the answer is. At the moment, we have an open question. Are microbes relevant in subsurface underground hydrogen storage? Well, some people say a lot, some people say a little. Some experiments suggest they may remove a lot of hydrogen. Some experiments, like the one done by Gabriela here, we will show them tomorrow, they say probably not, unless we stimulate the community with other things. But we have no definitive answer. And so you should worry because it's an unknown right now.
Is there a way that we can attack this problem by considering the worst case scenario?
That's what people have been doing a lot. The worst case scenario means I get brine from a reservoir and I give them everything they need to do their worst. I give them 100 millimolar sulfate, glucose, excess hydrogen. Okay, you have the worst case scenario. Now, are you gonna pump sulfate down your reservoir? Then when you measure the brine as 0? I doubt. I mean, it's a reservoir where you know sulfate is almost 0 because they are on land. There are not layers that can provide sulfate. There are not gypsum layer and things like that.
So it has to be constrained within the reservoir that we.
Of course, the worst case scenario is that your microbes will eat all the hydrogen, produce it to S, corrode your system, precipitate and clog your pore. Here we go. Worst case scenario. What are we gonna do with the worst case scenario? I mean, it's good to have end member of possible activities, but at the end of the day, your reservoir is not an end member. Sitting somewhere
I
along there.
suspect that most of the recent experiment studies are all polarized on the worst case scenario.
Yes.
Which I suspect they don't represent really what's happening.
What we did run with Gabriela and Martina has been a more neutral case scenario. Let's not go crazy enough. In some, we did a lot of replicates, you'll see tomorrow. In one of them, we did add sulfate, but the other, we did not. So let's see. In this reservoir, there is no sulfate. What's the need to give sulfate? To demonstrate that can be used by microbes? Whatever you give, it can be used at one point or another. So we need to be realistic. And what we need, sure, we need end members, worst case, best case, for a subset of things. But we need to know also the rules that connect the end member, right? Is it a simple linear mixing between using and not using sulfate, producing and not producing H2S? Is it complex mixing lines that have boundary point where things don't happen and then they suddenly happen a lot? We need to learn these rules in order to be able to make prediction. We're never going to be able to do every single situation, right? We need to go back to first principle. First principle means get end member and start to understand how the processes behave along those variables. Yes.
Sorry, but I don't think it's that approach to redefine the worst case scenarios in this case, because of course it's better to know all the process what's going on, but it will take a while. First, I think it's better to define worst case scenarios and their probability, having those.
No, I agree.
How much hydrogen is going to be lost because of microbial activities, because of the geochemical, and because of this absorption and some leakage pathways? And is it economical to operate this field after?
Yeah, but that field, worst case scenario for microbiology in this case is, I'll give you a geochemical example. You do your sand from the reservoir reaction with hydrogen in the lab. Okay. Worst case scenario for you would be added a ton of nickel and increasing the temperature to 500 degrees to demonstrate the nickel can catalyze the hydrogen oxidation under those conditions. Do you have nickel in your reservoir? No. Are you at 500 degrees? No. So what type of worst case scenario are you simulating?
But the nickel concentration of the one that found in some of the reservoirs would be a better start.
Could be a better start, but what we do in Gofen right now is we go into Germany to one reservoir, getting the brine, and saying, oh, there's no sulfate here. What happens if I give them a lot of sulfate? Worst case scenario. Sure, but we are in a hypothetical. We've been doing a lot of hypotheticals right now, okay? Knowing the caveats, there are laboratory tests, meaning that very often microbes are not tested at in situ pressure. There are a few laboratories that have been doing, but not a lot. Many are run at 2 bar, at 3 bars. We know things are different, right? Hydrogen solubility will be different if you do 50 bar against 2 bar. And so we are getting a lot of these end members, but then we are concluding that, oh, we should worry a lot about sulfate reduction because of the worst case. I don't agree with that. I agree we need the end member. We are on the same page on this. We need to start somewhere, and end member is a good start. But at one point or another, we need to be realistic about where we sit with the reservoir. Because if you say, is it economically feasible to run this reservoir under the worst case scenario? What worst case scenario are we talking? Are we talking about hydrogen consumed by that community there? Hydrogen consumed by that community if we pump down 500 millimolar of sulfate? That community there with 5 millimolar of nickel chloride as a catalyzer, you can make it as worse as you want, right? So let's consider boundaries where we are. I doubt you run your geochemical experiment for average Europe reservoir, adding 20 times what they have in nickel inside, because it will, you're just looking for the highest hydrogen reactivity at that point. Is it relevant? No. Is worst case scenario? Yeah, it's one of the worst you can find, maybe somewhere. So when we give to the community sulfate, acetate, glucose, hydrogen, we are very far away from in situ condition. And we need also that. But what I'm trying to convey here is that complex systems at one point or another require you to approach them as such, right? Not single variable, also because variable interact. So I'm not saying they're bad experiments. I never said that. I'm saying we've been doing those, but it's time we move beyond those into things that are more relevant for real condition. Because otherwise, right now, everybody that touches a microbe, there are possible sulfate reducers. So please do not inject anywhere, because in the worst case scenario, microbe can use it.
Which is not true, because we injected stuff in the carbon, right? But let's see, there's like in the field and the Schnurwit, where they're injecting the CO2 science here, so it's not disappearing or whatever, so we have the complex.